Oh, sorry, I see you already there. Nice to see you. So, wait, let me get something. Today we will have a look at viral replication and especially how viruses reproduce using the Lytic and the Lysogenic cycle. So stay curious and enjoy! But first, take a glass of milk, sit down and enjoy because we will talk about the discovery of viruses. Oh, we should start. Wait. It started all with tobacco mosaic disease. It infects tobacco plants and reduces yield during harvesting. The plant does not die, but growth is stunted. The leaves of an infected plant look like this. They show discoloration which may look mosaic-like, like you can see here. Adolf Meyer discovered in 1883 that he could pass on the disease from one plant to another by rubbing the sap of an infected leaf onto an healthy plant. He concluded that the disease is passed on by a small bacterium which cannot be seen under the microscope. Dmitry Ivanovsky had the idea to filter the sap using a bacterium filter a decade later. However, the disease still passed on and he concluded that either the bacterium is too small or it produces a filterable toxin. This was ruled out by the Dutch botanist Martinus Beijerinck. He discovered that the disease does not cultivate on a petri dish, which it should be able to if it was a bacterium. In 1935, the American scientist Wendell Stanley crystallized the infectious particle and could see it under an electron microscope. This was the first discovery of a virus. So that's it for the history part of this video. Put your coffee to the side and let's explore what viruses really are. So let's talk about phages, but what are phages? Well, phages are a type of virus, and they are not the only type of virus. There are many different viruses. When you think of the term virus, you probably think of something dangerous. Something which attacks your body till it gets ill. Like an army trying to capture a big castle and letting the people which live in there suffer. This is not really cool, but it will have an immediate impact on the castle itself, like viruses can have on your body. Luckily, not all viruses are harmful, thanks to our immune system. Here are some viruses. Retroviruses, adenoviruses, and bacteriophages. These viruses are pretty similar, but even though they have little differences which are important. So what are these exactly? Let's look at them in detail. Adenoviruses are non-enveloped viruses, which means that they do not have a lipid bilayer. Instead, they just have proteins called capsomeres, which form a coat called capsid around the genetic material of the virus. Adenoviruses have a double-stranded DNA genome. Retroviruses are envelope viruses. In contrast to the adenovirus, they have a lipid bilayer in addition to the capsid formed by capsomeres, protecting the genetic material. The envelope also allows the virus to enter cells by fusing with the membrane. The origin of the membrane is often the membrane of a host cell. This process is called budding or shedding. So the viruses just use the membrane of the host cell to form their own viral envelope. The genetic material of a retrovirus consists of two identical single-stranded RNA molecules. Last but not least, let's look at bacteriophages. Bacteriophages will be important later, so try to focus and memorize. They consist of a head and a tail. The head encapsulates the genome. The tail consists of the neck and the collar, the tail sheet, the base plate, and tail fibers, which are all made up of proteins. The tail fibers are flexible structures that can change their shape. Since bacteriophages are made up of proteins and do not have a lipid bilayer as a membrane, they are non-enveloped viruses. They may encapsulate a double-stranded DNA genome, a single-stranded DNA genome, a single-stranded RNA genome, or even a double-stranded RNA genome. Now we had a brief look at some types of viruses, but be aware that these three I mentioned are just some examples. There are many more virus families and different subfamilies. You saw that scientists can separate viruses into two main categories: the envelope viruses and the non-envelope viruses. An example for an envelope virus would be the retrovirus, while an example for a non-envelope virus would be the bacteriophage. You also must keep in mind that viruses are much smaller than bacteria. Interestingly, bacteria count as the smallest living organisms, while viruses are defined as non-living. When you look at numbers, a virus is about 20 till 100 nanometers big, compared to an animal cell which is around 10 till 100 micrometers big, while a bacterium is about 1 till 4 micrometers big. This is really small. Maybe it helps to think of a house being an animal cell. A bacterium would then be the size of a human, and a tennis ball could be a virus. We learned two things now. First, the structure of viruses, and second, that there are many different types of viruses. But what cells can a virus actually infect? There are viruses that can infect plant cells, animal cells, or even bacteria. This means this little guy can also suffer from the attack of a virus. However, it differs depending on the virus. A virus may have one specific target it wants to affect, or one specific cell type, like a blood cell or a muscle cell. This is called the host range. There's also a lock and key fit model between the receptors of a cell wall or cell membrane and the proteins of a virus, which means that not every virus can bind to every cell. This is also the reason why there are many different diseases. There are many different viruses which infect different cells. So when you have a disease, or more specifically a virus infection, your symptoms depend on the virus which attacks your body. That's also the reason why there are diseases which only affect plants. These specific viruses can only target plant cells but not animal cells. Bacteriophages, for example, target bacteria. So far we covered the basics we need to know to talk about the lytic and the lysogenic cycle. But you may ask yourself now, why is there a lytic and lysogenic cycle used by bacteriophages? Can't they just do mitosis like animal cells? Well, like I mentioned earlier, viruses only consist of proteins, the genetic material, and sometimes a lipid bilayer. Viruses are defined as non-living organisms because they cannot reproduce by themselves. They lack ribosomes or any other organelles which animal or plant cells have and are necessary for independent reproduction. Therefore, a virus needs a host cell to reproduce. And that's what we want to have a look at now. How do bacteriophages reproduce using a host cell? I asked you to memorize earlier, but let's recapitulate quickly. A bacteriophage consists of a head, which contains the DNA genome. In this case, a double-stranded DNA genome. The neck and the collar, the tail sheet, and the tail fibers. Keep in mind that the tail fibers can change their shape. This will be important now. During the first step of infection, the bacteriophage attaches to specific receptors on the membrane of a bacterium. Let's take an E. coli cell as the example host. After binding, the tail fibers of the bacteriophage are able to change their shape so the tail can move closer to the membrane and release the DNA from inside the viral head into the E. coli cell. At this point there are two ways in which the reproduction can continue. Either the Lytic or the Lysogenic cycle starts. First, let's have a look at the Lytic cycle. The distinguishing fact is that it culminates in death of the host cell. The viral genes immediately turn the host cell into a production factory. First, the virus uses the enzymes of the existing bacteria to replicate the viral DNA. Take care, prokaryotes like our E. coli cell do not have a nucleus. The DNA is just floating around in the cytoplasm, and so are the enzymes. By transcribing and translating the viral DNA, the host cell produces viral proteins without even knowing. One of the first transcribed viral genes leads to an enzyme which hydrolyzes the host cell's DNA, which then cannot be used anymore. Keep in mind, this is all done by the enzymes of the host cell themselves. The virus takes full control of the host, which is kind of barbaric. It's like when someone walks into your house and says, "Everybody will do now what I say." This will probably end up with you cleaning the kitchen, which is not that cool. This process takes 20 to 30 minutes, till finally the bacterium lyses. That means the plasma membrane breaks open with the help of lysozymes, another type of enzyme, letting the produced phages loose. The produced phages can now move freely through the body and infect other cells, which then serve as the new host cell. A phage that only reproduces by the Lytic cycle is called the virulent phage. You probably wonder why not all bacteria have died out if viruses easily make them explode. On the one hand, bacteria are not defenseless. Some bacterial mutants have receptors which no longer recognize particular phages. Also, bacteria have restriction enzymes whose job it is to cut up foreign DNA of a virus. The phages and the bacteria are in a constant evolutionary flux. This means that the phages may mutate, making the bacteria defense system useless, but then the bacteria may adapt again. This struggle for survival was described by the driver of evolution Charles Darwin 100 years ago. This dude, a real genius. But bacteriophages do not always have to infect the host cell in a way that immediate production of phages is followed directly. In the lysogenic cycle, the phages infect the cell and inject their DNA. So far the process is the same, but now the host cell does not directly produce the viral proteins needed to build phages. Phages that use this strategy are called prophages. After infection, the genome of the prophage is integrated into the bacteria's DNA. One prophage gene encodes a protein that prevents the E. coli from transcribing the other viral genes. As a result, the DNA of the prophage is mostly silent. When a cell now divides, it also passes copies of the DNA of the prophage onto the daughter cells. So even though the cell is infected, the viral genes are silent. The viral genome is kept and replicated until a switch to the Lytic cycle occurs at random. This switch is caused by environmental signals such as radiation or the presence of certain chemicals. Switching into the Lytic cycle will then again culminate in death of the host cell. There are also phages which reproduce by both cycles, the Lytic and the Lysogenic cycle. These phages are called temperate phages. They can choose between both cycles after attachment. Now, let's summarize again. First, the phage attaches to the bacterium. Now, the DNA of the phage is injected. The phage detaches, and from this stage on, either the Lytic or the Lysogenic cycle starts, depending on the phage. Let's have a look at the Lytic cycle first. The first step of the Lytic cycle is the degradation of the host cell's DNA. Afterwards, the synthesis of viral genomes and proteins starts. The viral genomes and proteins are then assembled to functioning phages. The last step of the Lytic cycle is that the membrane breaks open, letting the produced phages free. Now, let's have a look at the Lysogenic cycle. The first step after the DNA of the phage is injected is that it integrates into the host's DNA. The bacterium now reproduces, which results in many bacteria containing the DNA of the phage. From there on, the viral genome is kept and replicated until a switch to the Lytic cycle occurs at random. Since scientists have known how the reproduction cycles of viruses work, they can use this knowledge to our advantage and apply it, for example, in biotechnology or gene therapy. If you don't know what gene therapy is, here's a short definition. Gene therapy is an experimental technique used to treat or prevent diseases. The idea is to use a virus as a transport vehicle. You can inject a specific gene which can not only make it harmless for humans, but also can lead to positive results. Imagine you're missing an enzyme in your body, and without that enzyme you have a certain disease and feel terrible. Now a gene can be injected into a virus which then encodes that enzyme. The virus can then infect a cell in your body with this gene. As a result, the cell in your body is able to produce the specific enzyme which is strongly needed, making you feel much healthier and better. Awesome! This was a short explanation of how viruses could be used, but let me give you a real example of how a virus can help doctors to cure disease. Let's look at sickle cell anemia. Sickle cell anemia is a recessively inherited disease. It infects hemoglobin, which is the oxygen-carrying protein in your blood cells. The cause is a mutation in the HBB gene. Due to this mutation, the hemoglobin proteins have a different shape. Our blood cells are full of hemoglobin, and cells carrying the mutated proteins look like this. They are not perfectly round as in healthy people, but elongated and shaped, well, like a sickle. Since form-fit functions, the sickle cells cannot fulfill their functions properly. They may stuck in the bloodstream and block the blood flow. This may lead to pain. Or even worse is that they carry oxygen less efficiently. And without oxygen, our organs, the muscles, the brain, just everything can't work. A treatment for this disease is bone marrow transplantation. However, for this you need a healthy donor, which is incredibly difficult to find, because only 1 in 1 million may have matching stem cells in the bone marrow. This is where gene therapy steps in. After the bone marrow of a patient is extracted, a modified virus is used to deliver a copy of the HBB gene into the stem cells. The own bone marrow of the patient, now containing healthy stem cells, will then be infused back into the patient, and they can produce working hemoglobin and normal round red blood cells. This means you don't have to search and wait until a suitable donor is found. This can save people from suffering and even save their life. This method is still in research, but the first success was reported in March 2017 when a boy in France was treated successfully. So, that's it. I hope you understood at least a little about how the Lytic and the Lysogenic cycle work, and how viruses can help us. If you want to watch a specific part of this video again, look in the description down below. If you want to watch more videos, feel free to check this or my other channels out. And I wish you a wonderful day, stay awesome, till the next time. Goodbye!